Okay, let's unpack this. We are diving headfirst into, well, frankly, one of the trickiest bits of modern medicine, getting these incredibly powerful new drugs exactly where they need to go in the body.
Yeah, it's a huge challenge.
Specifically, we're talking about siRNA today. Those amazing molecules that can basically tell a specific gene to quiet down. Designing them, that's getting pretty good.
Right.
But delivering them effectively,
Yeah.
safely, that's the whole game, isn't it?
It really is. It's the bottleneck.
And our source for this deep dive is a bit different. We're not looking at just clinical trials or basic science today. We've got a detailed paper looking at the patent landscape for therapeutics siRNA delivery.
That's right. And patents, they're more than just dense legal text. When you analyze thousands of them, like this study did, they become a kind of map.
A map? How so?
Well, they show you where the innovation is happening globally, who's investing heavily, which technologies are buzzing, and really what problems the whole field is wrestling with. So for anyone tracking biotech, looking for opportunities, or just trying to grasp the strategy here, understanding this patent landscape is, well, it's crucial. Absolutely essential stuff.
Right. So our mission today is to basically navigate this complex map for you. We want to zero in on the key delivery technologies, figure out who the big players are from the patent filings, and understand what it all means for actually getting these therapies to patients and, for biotech strategy overall.
Sounds like a plan.
All right, let's just quickly level set on the basics. For listeners who follow biotech but maybe aren't deep in the molecular weeds, what exactly is siRNA and why is delivering it such a headache?
Okay, sure. So siRNA stands for small interfering RNA. Think of it as a tiny instruction molecule. Its job is to tell your cells to switch off a very specific gene, maybe one that's causing a disease.
How does it do that?
It gets incorporated into this cellular machine called the RISC complex. RISC is like a guided missile system for messenger RNA.
Messenger RNA being the blueprint for making proteins.
Exactly. The siRNA acts as the guide, telling RISC, find this specific mRNA blueprint. And then RISC, well, it chops it up, destroys it.
So no blueprint, no protein. You silence the gene.
Precisely. You stop that specific protein from being made.
The potential seems massive. I mean, if a disease involves a problematic protein, you could just turn down the volume on the gene making it.
That's the promise. It's huge. Relatively easy to design for almost any gene target. Highly specific. And it's not just theoretical anymore.
Right. There are approved drugs.
Yes, we have approved siRNA therapies already helping patients with conditions like hereditary polyneuropathy, acute hepatic porphyria, a rare kidney stone disease called primary hyperoxaluria, and even high cholesterol. So the principle, proven. It works.
Okay, so where's the catch? Why the thousands of patents just on delivery?
The catch. It's a big one. Getting that siRNA molecule safely from, say, an injection all the way into the right cell and then to the right place inside that cell, it's incredibly difficult.
What makes it so hard?
Well, first off, the siRNA molecule itself is quite fragile if you just inject it naked into the bloodstream.
Uh-oh.
Yeah, exactly. Enzymes in your blood. These nucleuses, they just chew it up really fast.
Yeah.
Gone.
Okay. Hurdle 1.
Hurdle 2. Your immune system often sees it as foreign, something unwelcome, and clears it out quickly.
Makes sense.
Hurdle 3. Even if some survives, just getting enough of it to accumulate in the specific organ or tissue you're targeting, that's tough. Biodistribution is a major challenge.
So survival, evasion, and location.
And we're not even inside the cell yet. Hurdle 4 is actually getting the target cell to take it up efficiently.
Right.
Crossing that cell membrane isn't easy for a molecule like siRNA.
Right.
And then hurdle 5, the final boss, maybe, even if it gets inside the cell, usually gets trapped in these little bubbles, these compartments called endosomes.
Okay.
But the RASC machinery, the thing it needs to work with, that's out in the main part of the cell, the cytoplasm. So the siRNA has to escape the endosome. And this endosomal escape, it's notoriously inefficient, a major bottleneck.
Wow. Okay, that's quite the obstacle course. No wonder there are over 11,000 patent documents just focused on delivery. That's a staggering amount of effort.
It really underscores how critical delivery is.
So what did the paper find about the overall trends? Who's tackling this? Where's the activity?
The big picture from the patent data is clear. This is a global race. Innovation and investment in siRNA delivery have just surged over the last 2 decades.
And who's leading the pack?
Looking at the distinct patent families, about 3,300 of them, the U.S. is way out in front. Dominant, owning or originating something like 51% of these patents.
half the field, basically.
Pretty much. Other key players are there, of course. Japan has about 9%, mainland China around 7%, Canada 6%, and others. But yeah, the U.S. is really the powerhouse in terms of filed patents.
Interesting. And who's filing these? Is it mostly big pharma trying to build pipelines, or universities and smaller players involved too?
It's actually a mix, which is common in cutting-edge biotech. Private companies hold the biggest share, about 54%, which makes sense, right? They're focused on commercializing therapies.
Sure.
But universities are major players, too, holding around 29% of the patents. And research institutions have another 13%. Importantly, the paper notes that university involvement has really grown over time.
So strong academic research feeding the pipeline.
Exactly. And you see a lot of collaborations reflected in the patents, too. University-industry partnerships, University Research Institute tie-ups, that kind of thing. It takes a village, really.
You mentioned mainland China earlier. The paper pointed out something about their patent families tending to be smaller. What does that suggest strategically?
Right. A patent family is basically the same core invention filed in multiple countries. A smaller family size generally means the inventors are seeking protection in fewer places globally.
So maybe more focused on the domestic market.
It could suggest that, yeah. Or perhaps just a different strategy regarding international intellectual property compared to, say, big U.S. or European entities who often file very broadly across all major markets to secure their commercial interests worldwide. It's a subtle clue about maybe the scope or the intended reach behind the patenting effort.
Fascinating, reading the strategic tea leaves in patent filings. Okay, let's get into the tech itself. What are the main strategies people are patenting to overcome all those delivery hurdles you laid out?
Broadly, the paper groups them into two big categories. First, you've got strategies that involve putting the siRNA inside some kind of protective vehicle or carrier.
Like putting it in a microscopic delivery van.
Kind of, yeah. These carriers can be based on viruses, though that's less common for siRNA now, or more often non-viral materials like lipids or polymers.
Okay, carriers. What's the second category?
The second approach focuses more on the siRNA molecule itself, modifying its chemistry to make it more stable or less immunogenic, or attaching things directly to it, like a targeting molecule, to guide it to the right cell. Sometimes this is combined with physical methods, too.
Like electroporation or ultrasound.
Exactly, using physical force to help get it into cells, though those are patented less often than the carrier or molecular modification approaches.
So based on the sheer number of patents, where's most of the action? Which strategies are drawing the most innovation, or at least the most patent filings?
Non-viral carriers are definitely the most heavily patented area. Within that category, two types really stand out. Lipid-based carriers, especially lipid nanoparticles or LNPs.
LNPs, right. We hear a lot about those.
Yep. They account for about 20% of the patent families. And polymer-based carriers are right up there too, also around 20%.
Okay, lipids and polymers leading the non-viral charge. What else is significant?
Biocarriers are surprisingly prominent, around 18%. These are things like extracellular vesicles, EVs, which cells naturally release. People are trying to engineer them as delivery vehicles.
Interesting, harnessing natural systems.
Then you have ligand siRNA conjugates attaching that targeting molecule directly at about 10%. And viral vectors like adenovirus are down around 6%.
It's notable that non-viral methods dominate so much, especially compared to gene therapy where viruses are still major players. And you mentioned combining technologies is a trend.
Absolutely. The patent landscape shows a clear move towards more sophisticated multi-component systems. It's not just lipids or polymers anymore.
What kind of combination?
You see patents for things like putting chemically modified siRNA into an LNP carrier, or creating hybrid nanoparticles that blend lipids and polymers, trying to get the best of both worlds.
So trying to tackle multiple hurdles at once with one system.
Exactly. Protection, targeting, cell uptake, endosomal escape. Researchers are layering functionalities. It shows the field is maturing, moving towards more complex engineered solutions because the problem itself is so complex.
Okay, let's dig into the strategy behind these. From an investor's perspective, or for someone watching clinical developments, what are the key strengths and weaknesses, the pros and cons of these main approaches?
Great question. Let's break down the big ones.
Start with LNPs, the lipid carriers.
Right, lipid nanoparticles, LNPs. They're a huge strategic advantage. Clinically validated. They're used on Onapatro, an approved drug. That's massive proof of concept. They're generally versatile, biodegradable, reasonably biocompatible. The challenges, and where the R&D is focused now, are things like long-term stability, potentially triggering immune responses, and crucially, getting them to efficiently deliver their cargo outside the liver. They naturally tend to accumulate in the liver.
So the strategic push is beyond the liver.
Definitely. Improving targeting to other organs is a major focus for LNP innovation. It's a proven platform, now being refined for broader use.
Okay, what about polymer carriers?
Polymers offer potential advantages in stability and, importantly, helping with that endosomal escape problem. The proton sponge effect is a mechanism some polymers use.
Sounds useful. What's the downside?
Toxicity. That's the big strategic hurdle. Some polymers can cause non-specific side effects. So the R&D game here is all about designing polymers that are really effective at delivery but have much better safety profiles. The tricky balancing act.
Makes sense. Then there are the ligand-siRNA conjugates like GalNAc.
Yes, the GalNAc conjugates are the prime example here, also clinically validated in drugs like giflurare and LQVO. The strategy is elegant. Attach a molecule, GalNAc, that binds specifically to a receptor found mainly on liver cells.
So targeted delivery without a bulky carrier.
Exactly. That simplifies things and can reduce carrier-related toxicity. But the limitation is obvious. You need a suitable receptor, highly expressed on your target cell. GalNAc works great for the liver because the ASGPR receptor is abundant there.
Not a universal solution then.
Not at all. Plus, receptor uptake can get saturated, limiting how much drug you can deliver. And even without a carrier, there could be toxicity concerns related to the conjugate itself. Some liver effects have been noted with GalNAc at higher doses or frequencies. Strategic takeaway. Brilliant for targets with the right address, like the liver, but receptor availability restricts broader use.
What about those biocarriers like EVs? You said they were getting significant patent activity.
Right, extracellular vesicles, EVs. The excitement here comes from them being natural packages that cells use to communicate. They're potentially very biocompatible, might evade the immune system better, and could potentially cross barriers like the blood-brain barrier. High potential.
But they sound early stage.
Very much so. Strategically speaking, the massive challenge is manufacturing. Getting pure, consistent batches of EVs at scale is hard, and reliably engineering them to actively target specific tissues rather than just going where they naturally go. That's still a major work in progress. Strategic takeaway. Watch this space. Huge potential, but needs breakthroughs in manufacturing and targeting engineering.
And quickly, viral vectors.
Efficient delivery, yes, that's their strength. But for siRNA, the safety concerns, immune reactions, potential integration into the host genome plus complex manufacturing generally make non-viral options more attractive. They dominate more in traditional gene therapy delivering DNA. Less of a focus for siRNA delivery strategy right now.
Okay, that really clarifies the strategic landscape. It's about matching the tech to the target and navigating those pros and cons. The paper also looked at who owns patents in which areas, right? And how patents connect.
Yes, they analyzed the top patent holders.
Yeah.
To show specialization, you see companies like Arbutus Biopharma heavily invested in lipid carriers, which makes sense given their history.
And Alnylam, they have approved drugs using both LNPs and conjugates.
Exactly. Alnylam, a clear leader, has strong patent portfolios covering both chemical modifications and lipid carriers, reflecting their foundational work. Companies like Roche and Arrowhead Pharmaceuticals are notable players in the ligand conjugate space. Universities often focus on newer materials or mechanisms.
And the connections between patents.
The citation analysis was revealing. Patents related to lipid-based delivery are cited very heavily by later patents across the board. It suggests LNPs have been a really foundational technology that much of the subsequent innovation has built upon or reacted to. It highlights their central role in the field's evolution so far.
You mentioned targeting beyond the liver as a key strategic focus. Is the patent activity reflecting that push?
Oh, absolutely. That's the next frontier. Extra-hepatic delivery. Getting siRNA effectively to places like the kidneys, the central nervous system, the eyes, muscles, that's where many unmet needs lie.
Much harder than the liver.
Generally, yes. The liver has unique features that make it easier for things like LNPs and GalNAc to get in. Targeting other tissues requires different strategies. And the patent landscape absolutely reflects this. Lots of filings describing novel carriers, targeting ligands, or formulations specifically designed to reach these non-liver tissues. Solving extrahepatic delivery is arguably the biggest strategic goal for expanding siRNA therapeutics.
So zooming out, we have 4 approved siRNA drugs, mostly hitting the liver using basically 2 validated delivery approaches, LNPs and GalNAc conjugates. But you said there are thousands of patent families covering delivery. Where does that leave all that other patented tech?
It's a really striking point the paper makes. Those 4 approved drugs relate to maybe 5 or 6% of the total patent families studied.
Only 5%. Wow.
Yeah. It means the overwhelming majority of these patented delivery innovations are still stuck in R&D, far from patients. It highlights two things. One, just how difficult it is to translate a promising lab technology into a safe and effective approved medicine. And two, the sheer potential that's still sitting in the pipeline. There's a vast reservoir of ideas out there.
But there are things moving forward in clinical trials.
Yes, definitely. There are over 50 siRNA therapies currently in clinical trials. But here's the key point for strategy. The vast majority of those trials are using the same workhorse technologies as the approved drugs, LNPs and GalNAc conjugates.
So companies are mostly betting on the proven platforms.
For now, it seems the main strategy is to refine and expand the use of those validated methods, applying LNPs and GalNAc to different disease targets, maybe pushing them towards slightly different tissues where feasible, rather than taking big clinical bets on completely novel, unproven delivery systems just yet. It's about optimizing the known quantities while the newer tech matures in the background.
Okay, so looking ahead, based on this patent map, what's the forecast? What should we expect to see in the coming years?
I think you can expect continued intense activity. The push for effective extra-hepatic delivery will absolutely dominate R&D strategy. We'll see a relentless search for better materials, safer polymers, more sophisticated lipids, maybe breakthroughs in those biocarriers like EVs. More precise targeting strategies will be key.
The underlying potential is still driving it all.
For sure. siRNA itself has inherent advantages. The speed you can potentially design new drug candidates, the specificity, the broad range of possible targets. The huge investment reflected in these patents shows a powerful, sustained belief in the future of this modality. Companies like Alnylam are clearly pushing beyond the liver into areas like eye and CNS diseases. The patent landscape paints a picture of a field that's had major successes, but feels like it's still just scratching the surface of its potential.
Wow. What a fascinating tour through the legal and strategic underpinnings of siRNA delivery. It really drives home that this isn't just a scientific puzzle. It's a massive arena for innovation, competition, and high-stakes strategy in biotech.
Absolutely. We've seen this incredible variety of technologies being patented: lipids, polymers, conjugates, EVs, chemical tweaks, each with its own story, its own pros and cons that shape company decisions. And while liver delivery is the big clinical success story so far, the patents make it crystal clear. The next strategic mountain to climb is delivering siRNA effectively, safely everywhere else in the body.
That gap you mentioned, thousands of patents, just a handful of approved drugs, really tells the story. Huge challenges, but also just immense potential still waiting to be unlocked.
Exactly. A story of hard-won progress and vast future promise.
Well, hopefully, this exploration of the patent landscape gives you, our listener, a much clearer picture of the complex science and the really critical strategic thinking driving this incredibly promising field of medicine.
Yeah, it's a dynamic space to watch.
And it definitely leaves you with a thought to chew on. If siRNA holds the potential to silence almost any troublesome gene, and the sheer ingenuity we see pouring into these delivery patents means we are getting better and better at getting where it needs to go, what disease that seems intractable today might just become treatable tomorrow?